Hello everyone, welcome to this session. Our chapter is the transportation materials and living things, and today we're discussing about the blood grouping. We've started discussing this in our previous session. We saw the various groups of blood and how they can be classified. We discussed it with the clear shown table. So in this session, we'll dive into more understanding the blood types and the rhesus factor. So let's start with the what is the blood grouping. So we'll see the simple meaning of blood grouping. The blood grouping, this is just the process of determining an individual's blood type. So when you try to identify the person's blood type, is what we call the blood grouping. It is just a simple and inexpensive experiment to perform just to obtain the individual's blood type, and only a few drops of blood are required just to enable a person just to carry out the experiment to identify the person's blood type. Now, this kind of blood grouping is essential for the safe blood transfusions, and the safe blood transfusions are required, or they are just critical in medical emergencies and the surgeries. Now, let's see how blood typing is done, or just how blood grouping is done. Now let's see some steps here, and later on we will just see what kind of blood grouping we can get. Here, the first step, mix anti-A antibody serum with part of blood sample. Then second step, mix anti-B antibody serum with the remaining sample. Then the third step, observe whether the agglutination or the cramping occurs. Now the last step, just the pattern of agglutination indicates the kind of blood type. So that's it, just how you can see or how you can tell the kinds of these groups of blood by simply carrying out these kinds of steps. Now let's see the interpretation of the blood type Rhesus. Now, for example, determining the blood type A. So, take an example, maybe an individual is blood type A. Now, here's how that kind of blood type A was determined. Now, if blood sample is agglutinated by the anti-A antibody, but not agglutinated by the anti-B antibody, so that's the crucial thing you can note here, just to determine the person's, for example, the blood group A or blood type A. It means that it is the agglutination happens at the anti-A antibody. So that's what it means. Now, for this case, A antigen is present on the red blood cells, and the B antigen is not present. So that's when we can say that therefore the blood type is the type A. So that's clear in terms of interpretation of the blood type results, as we've carried out the steps that we've seen in the previous slide. Moving on. Now let's discuss about this, the rhesus factor. Some people have an extra antigen called the Rhesus antigen on their red blood cells, while others they do not have it. So this is the other factor that is there that it can contribute to the grouping of the blood. So some people they have this an extra antigen that is we call the Rhesus antigen on their red blood cells, while other people they just do not have it. Now the Rhesus positive, they have the RH antigen, while the rhesus negative they do not have the RH antigen. So you can have this RS factor as people who have it, and people who do not have it. So that's the other extra antigen that you can observe in the red blood cells. Now, location in blood, you can tell that the RH antigen it occurs in red blood cells, and the RH antibody just occurs in the blood plasma. So you can tell the location of the rhesus antigen and the rhesus antibody. The rhesus antigen it occurs in the red blood cells, and the rhesus antibody it occurs in the blood plasma. So rhesus factor in blood transfusion. So let's see how this rhesus factor it contributes to the blood transfusion. Now, if rhesus this RH antibody it mixes with the RH antigen during the transfusion, the agglutination will occur. Now, the RH positive blood can stimulate the Rh negative person to produce the antibodies against the Rh plus antigens. Now, the Rh negative blood cannot stimulate the Rh positive person to produce the antibodies against the Rh negative. So, therefore, from these explanations, you can say that an Rh positive person can safely receive blood from an Rh negative donor. So that's the extra factor that you have to consider in terms of the blood transfusion when we're observing the groups of the blood of the various individuals. So complete blood type notation. So the complete blood notation of individual, you can see having, for example, AB plus. So this is basically group AB rhesus positive. So this blood type, it indicates the following: both A and B antigens are present. So that's the group A, B, and also the rhesus antigen is present. That is the positive rhesus antigen, and written as AB Rh positive or simply AB positive. So I hope that's clear in terms of how the complete blood type notation or the complete blood group notation you can observe for a given person or individual. Now, key points summary. So we've covered up for this session. So the blood types uses the anti-A and the anti-B bodies. We said that the agglutination indicates the presence of the specific antigens. The blood groups AB, and O. So those are the blood groups. Rhesus factor, we have the rhesus Rh positive, we have the antigen, and the Rh negative, no antigen. The Rh positive persons, they can receive blood from the Rh negative donors. The complete notation, for example, AB positive, meaning the blood group AB rhesus factor positive. So that's all I have for this session. I hope that's clear, everyone. Thank you for listening. So this is all I have for this session, and see you in the other session. If you have any question resulting from this session, you can just leave it in the comments. So I hope that's clear, and thank you.